All right, everyone, thanks for coming to Laurel's thesis defense. We're so excited when this day arrives. It's a little bit sad, too, because it's always sad when you lose a good student. So I want to first start off by thanking the Ichthyology Lab for providing the spread afterwards. So if you have never had noodle burritos before, stick around. It's quite the delicacy. Carbs wrapped in carbs. So it'll keep you going for the rest of the day. I'd like to thank Laurel's family for coming, so her dad, Eddie, and her mom, Hannah. And then her significant other, Brendan, are here, so thank you guys for coming. I'd like to thank Laurel's thesis committee, so Geeta McDonald, Rick Starr, who's one of the world's experts on Laurel study species. It was great to have him involved. And then Jamil Samhori, who's at the Northwest Fisheries Science Center, so the NIMS lab up in Seattle. And he's actually here today, which was great. He gave a seminar at Hopkins on Friday, and so we convinced him to stick around for the defense. And the project you'll hear about, Jamil and I have known each other since we were in college. We've been good friends since then, lots of shenanigans and antics along the ways. But we saw this project as an excuse to spend more time together. Going out fishing is funny. We're two fish biologists, but we're probably the worst fishermen on the planet. And we got this project where you have to go fishing. So we needed to find people like Laurel and Bonnie Brown to really do the project for us, because we're terrible. So anyways, a little bit about Laurel. So Laurel and I have known each other for over 10 years now. So Laurel did her undergraduate at UC Santa Barbara, and I think it was the first year that I was teaching the ichthyology class after I had TA'd it for 6 or 7 years, however long that dissertation lasted. And Laurel was a student in that class, so I got to know her in the class. And then she also worked a little bit as a volunteer in the lab that I worked with, the Pisco lab. And then after Laurel finished UCSB, she moved up to the Bay Area. She got a job as a scientific aide with California Department of Fish and Wildlife now. She worked on the recreational fishery sampling projects, so she interacted a lot with recreational anglers, sampling their catch. She also worked on some of the commercial projects for salmon and for herring. And then she worked as well at the Aquarium of the Bay in San Francisco, educating the public about marine life. And at some point, she got the itch to come back to grad school, and we kept in touch a little bit over the years. And she said, "I'm interested in fisheries issues and fish ecology. Do you know any good places for that?" And I was like, "Well, why don't you come down and talk to me, talk to Rick Starr? It seems like that's what you're interested in." And she did so, and things went great. She submitted an application, and as I started reading her personal statement, it really grabbed me right from the get-go. I pretty much only needed to read the first line and knew that she was the perfect person for my lab. So let's see if you guys agree that this was a good fit for her. So Laurel said, if this works. Come on, computer. We'll never know. Maybe I need to stop it. Let me stop it, start it again. There we go. Okay. So here was the first one. I enjoy eating fish, fishing for fish, drawing fish, and reading about fish. She said fish 5 times in one sentence. And for me, all I heard was fish, 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 fish. And I was like, perfect. Yep. So let's see if she actually lived up to all of these things. Does she enjoy eating fish? Yep, there's evidence for that. She's cooked me a number of meals. Even when she's freezing, she's still dedicated to the task of filleting fish, getting them ready for the meal. Does she enjoy fishing for fish? Yes, definitely. As you'll hear about her research project, she spent about half the time here fishing for fish, not only for her project, but working on a number of other projects as well. What about drawing fish? Yep, check there. These were all examples of Christmas cards that she sent me even before she was a grad student here, telling me what she was doing. And she's a fantastic artist. Always nice puns in there, fish puns, which I loved. It was great stuff. Okay. And what about reading about fish? Well, you can't win them all. 3 out of 4, though. 3 out of 4. What she also said in her statement of purpose was about what she's interested in. I've been captivated with rockfishes, lingcod, cabezon, sculpins, and greenlings. I'm interested in species interactions and population dynamics, but I'm more curious about how they fit into California's commercial and recreational fisheries. I highlighted Lingcod up there because that ended up being her study species, and this is a pretty good synopsis of what she did for her thesis. She looked at fisheries issues, population, structure. You'll hear about all that today. So this is one of those examples where she pretty much did what she said she was really interested in doing when she came here. Right, so, but in the process of doing this project, I mean, who would have thought starting out here is this cute kid, right, this really accomplished, responsible Girl Scout, right, this loving daughter, right? Who would have thought that Laurel would grow up to become a serial killer, really? Yeah. Of fish, not people, right? Only for research, right? When she has the appropriate permits in place. So just a brief summary, kind of, Laurel has really accomplished a lot here in her little over 4 years at Moss Landing. She's really done an impressive amount of work, which you're going to hear about. So she worked on this project that we had funded from NOAA to study lingcod populations. Her and Bonnie spent 2 years sampling lingcod up and down the coast. They single-handedly organized over 50 fishing trips and all the volunteer anglers to participate. They caught over 2,000 lingcod, sampling from Alaska to Southern California. And she learned how to age these lingcod using all the standard techniques in the industry. She aged over 1,500, I'm just guessing, and maybe more than that, lingcod. She also just took the initiative to take a semester and basically move up to Seattle to take kind of the best fisheries science class there is, to learn how to program in R. While she was up there, her data that she collected was actually integrated into the lingcod stock assessment. It actually improved some aspects of that assessment. And so, I think there's very few students who can say my data before I even finished has actively gone in and it has affected fisheries management. I keep saying I'm doing research that does that, but I don't even know if I've ever really done that. So this is great. And this has led to these other extensive collaborations with NOAA fisheries scientists and taking the lingcod study in lots of different directions, which is great. A lot of service as well. So she was the student body president here at Moss Landing. Right? She did a lot for open house, including organizing and running the t-shirt design contest one year using her art skills there. She was the student representative for a couple years for the Marine Network, organizing a lot of those events that happened here at Moss Landing. She was a TA for our Baja class, and that helped us when we moved to a brand new location to get all the logistics and everything together. She won a lot of awards, so Simpkins Award, the Myers Trust, the CSU Coast Award, the Lowry Family Scholarship, International Women's Fishing Scholarship, the WAVE Award. So in addition to having some funding for her project, she also went out there and was very active in finding her own funding as well. She gave some great talks at meetings, the American Fisheries Society, Western Society of Naturalists, Western Groundfish. She mentored a number of UROC students on her project, and so she was just active in lots of different ways while she was here. But I think, maybe she did a little too much. And we sort of worried that, did we work her too hard in her master's thesis? Because half the time you look and she has a gift. She's sleeping somewhere, everywhere, no matter where you look. And so I'm thinking maybe she was so productive because she was so rested all the time, just so well rested. Oh my God. Oops, what happened? Where'd the last one go? Oh, it's freaking out now. Maybe I have to. Oh, there we go. And finally, here's one of my favorite ones. So here, her thesis committee member, Rick Starr, she's reading some literature about his research program. I think she was very interesting. So anyways, with that, Laurel Lam, just a very interesting creature, her and Rusty. So I can't wait for her to tell you about what she's been up to the past couple of years. So, Laurel.
Thank you for that introduction, Scott. I napped right before this, so don't worry. Okay. Let me get this ready. Okay. Should be open. All right. Let me see if the pointer works. Okay. So thank you all for joining me today. I will be talking about my master's thesis, which is looking at the geographic and habitat-based variation in Lingcod demography and life history across the US West Coast. So I've broken down my thesis into 2 chapters, and this will be the general outline. So for chapter 1, I'll be talking about Lingcod demography and life history variability. And then I'll be applying what we find in chapter 1 into chapter 2, where I'll look into the environmental correlates of life history and its implications for Lingcod stock structure. And this will be all wrapped up together in the conclusion and final management recommendations. So chapter 1. So marine fishes are exposed to unique fluctuating environments that are affected by oceanographic variability, habitat complexity, and human-induced impacts. Studies have found that broadly ranging fish species can exhibit interspecific variability in demography and life history traits. So meaning things like population structure and growth and maturity rates of the same species can be different depending on the environment that they live in. So for chapter 1, I'll be talking about the 2 main parts of chapter 1. I'll just go over the terms I'll be using a lot. So when I mention demography or population structure, what I'm referring to is the frequency of size or age classes in a population. So for example, you can see that population A has a higher frequency of old age classes, and population B has a higher frequency of young age classes. And then when I talk about life history, what I'm referring to are rates of growth, longevity, or how old the fish gets, size and age at maturity, which indicates the timing of maturity, and natural and total mortality rates, as well as condition, or overall health. So past studies have found that fish can exhibit geographic differences in life history. According to Bergman's rule, as latitude increases, so as we go into colder waters, we typically start to see larger sizes, faster growth, and later maturity. And this has actually been seen in terrestrial animals as well. This is generally attributed to the temperature-driven energetic trade-offs between somatic growth and reproduction, as well as regional variability in food availability and metabolism. So this pattern is seen in fishes, like I mentioned. Along the US West Coast, many species of rockfishes have been found to grow faster and larger in northern waters compared to southern waters. And in addition to geographic or latitudinal-based differences in life history, there can be differences based on habitat. So depending on the life stage, the same animal can use different habitats for different reasons. For example, younger age classes can benefit from living in a complex nursery habitat that helps kind of hide them in those vulnerable life stages, and so they can bulk up and get fat before moving into more open areas. And as adults, they can have strong habitat associations, such as this kelp rockfish hanging out in the kelp forest. Habitat usage can also vary across geographic regions due to differences in substrate availability, complexity, and distribution. So how patchy or continuous the habitat distribution is. So on the bottom, there's 3 examples of different kinds of habitats with varying complexity. So on the far right, you see a cobble or pebble sort of habitat that's pretty low relief compared to high rocky, like seamount, kind of a high relief, and then you have sort of a kelp forest, kelpy area. So fish living in one habitat can grow or live differently than a fish growing in another habitat, or than the same fish living in a different habitat. So why is this important? So why are we interested in how growth, maturity, and other life history traits can vary? Well, it's because it can have important implications towards how we manage fish populations, especially ones that are broad-ranging over different environments. So for commercially and recreationally exploited fish stocks, stock assessments are typically based on life history traits and are used to monitor how productive a population is, how to set regulations, and how much of the population can be harvested on an annual basis. So understanding the differences in life history can help increase the accuracy of these stock assessments. However, modern stock assessment methods typically assume that life histories are spatially constant. So we run into problems because stocks end up being managed over broad geographic areas with catch quotas, size limits, and other regulations applying equally in all places. So we run the risk of over-harvesting in one area while under-harvesting in another. So in order to address these limitations, we first need to understand the spatial scale at which life histories can vary. So this brings me to my study species, which is the lingcod. So lingcod are commercially and recreationally harvested across the coast, usually along with other rockfish species. They're also important ecologically as a top predator in the nearshore ecosystem and typically eats a variety of squid and fish, but will also bite opportunistically on wounded fish or hooked fish as well. They're very broad-ranging from Alaska to Mexico and are found over 2 habitat types, or various habitat types, but most often they're found over low relief areas or high relief rocky areas. And knowing that they have a relatively small home range and don't tend to move very much, this makes lingcod and other bottom fish ideal candidates to study for these life history variations. And while many studies on lingcod life history have been conducted in the past, they were done in relatively small areas, mostly in northern waters. They used different collection methods, and they were done over various time periods. So it makes those past studies kind of hard to compare. Now we'll give a little bit of background on the lingcod fishery. So the lingcod fishery is federally assessed on a regular basis, and lingcod are caught recreationally using hook and line gear and commercially using bottom trawl gear. And this is important to distinguish because these 2 gear types are essentially targeting lingcod over different habitats. So from the high relief areas, that's where the recreational fishery is typically targeting using hook and line gear. And then over the low relief habitats, we have the commercial fishery, and they're going over low relief areas. So since these gear types don't really overlap habitats, it's important to note that there's potential for life history to be different. So the lingcod stock assessment actually uses life history parameters that are based only on trawl caught samples. And those samples come from the West Coast Groundfish Bottom Trawl Survey. And this can be limiting because we're essentially excluding lingcod from those high relief areas. And so we're not accounting for the differences they might exhibit in growth, maturity, mortality, and population structure. So the objectives of my study for Chapter 1. My first objective is to quantify and compare lingcod demography and life histories across the US West Coast between 2 habitat types, low relief and high relief. So my predictions for what we expect to find, I predict that as latitude decreases, we'll find lower levels of, or lower rates of growth, younger ages, smaller sizes, and we expect lingcod to be maturing at smaller sizes as well, or at earlier times. And I also expect condition to be lower for lingcod in southern areas because warmer waters typically imply areas of lower primary productivity. And I expect higher natural and total mortality in southern regions since younger ages typically imply higher natural mortality rates. And between habitat types, I predict that growth, longevity, mean sizes, and timing of maturity will increase. Condition will increase in high relief areas since high relief areas can support a higher abundance of fish, invertebrates, and other potential prey items for an opportunistic predator like lingcod. And because we expect them to live longer and have higher longevity, we'd also expect their natural mortality rate to be lower. And because the recreational fishery is larger than the commercial fishery at this time, I expect that the total mortality will be higher as well. So going into my methods. So lingcod were collected from 7 regions across the US West Coast, went from Alaska all the way down to Southern California. And 2 to 4 ports were chosen within each region. And the colors on this map will be kind of corresponding to the colors I use in my figures. So if we see a good latitudinal gradient, we'll see this awesome rainbow pattern. So across the coastline here, there is a large gradient in sea surface temperature. So colder waters up north and warmer waters down south. The California current that runs along the coast controls much of the productivity and upwelling in these coastal regions. And since colder waters typically imply higher productivity, regions in the northern California current are typically higher in primary productivity than they are in the south. And again, the 2 habitat types of interest that we researched over were high relief rocky areas and low relief trawlable habitats. So in order to collect Lingcod samples from high relief habitats, we chartered party boats from 26 ports along the coast. So we worked with charter boat captains to find optimal fishing areas that are typically targeted by the recreational fishery. And any bycatch that we caught, any rock fishes, would be identified, measured, and then descended back to depth. Couldn't edit that part out. Some of the totals from our hook and line sampling. So we used one RV over 2 years, had 3 amazing undergraduate interns, worked with 22 boat captains, had almost 300 volunteer anglers, spent 70 days at sea, and landed 2,189 angry lingcod. So the volunteer anglers that we had were really the backbone of this project because we couldn't have caught any fish without them. So it was really important to us that we had volunteer anglers that really knew what they were doing and were knowledgeable about fishing. So, you know, can't win them all. And across the trawlable habitats, we had some help from the West Coast Bottom Trawl Survey. So this is an annual standardized survey that's been going on since 2003. And the survey, this trawl survey is the primary source of fishery independent data for the West Coast stock assessments. And it collects federally important groundfish species along the upper continental shelf and slope. It's typically targeting depths of 55 to 1,000 meters and runs from the US-Canada border to the US-Mexico border. And it's reflective of the areas and habitats that are exploited by the commercial fishery. So here's a quick catch summary table of what the samples we got from each habitat type, and the, you know, kind of be mindful of the locations. We only got high relief, or we only got trawl samples from Washington to Southern California. So something I wanted to highlight is that the trawl samples were collected at much deeper depths than the hook and line boats could access. So for my analyses, only depths less than 555 feet were used. I'd initially ran all the analyses using Lingcod from only the overlapping depths, but because the results were the same as when I included the shallower depths, I chose to keep the shallower depths as well. And between the sexes, high relief lingcod were predominantly male, and majority of the low relief lingcod were female. I won't be talking too much about sex-based differences in lingcod today, but you can read more about it in my thesis. But I just want to point out that this ratio was pretty consistent within each region. So for all of my subsequent analyses, only depths less than 555 were used, and the sexes were pooled. So after we collected the lingcod, we would bring them back to the lab. All samples would be measured, weighed, and fully dissected. We collected genetic samples and stomach contents for a diet analysis. All the organs were weighed as well. And a big majority of my lab work consisted of aging lingcod. So lingcod are aged using the second dorsal fin from rays 4 through 8. It's very specific. So the fin ray section is first dried and then hardened, and then cut into sections using that high-speed saw. And then they're cut into sections, mounted onto slides, and are ready to be read under a microscope. So much like how you would count tree rings to age a tree, here you would need to count the number of annuli, or the little white marks that indicate that form once a year during winter growth. And special thanks to the Washington Department of Fish and Wildlife for teaching me how to prep the fins and for teaching me the proper age reading process. So as a method of quality control, I did a blind read of all the fins once to see if I showed any aging bias with myself, which I did not. And then I also compared my age reads to the Northwest Fisheries Science Center Aging Lab in Oregon and to the age readers at the Washington Department of Fish and Wildlife to test for interlab bias and accuracy. I found that my age reads were within the accepted range of aging error, which is less than 7.6%. And for the rest of my life history analyses, I compared size and age structures between populations, between regions and habitats. And again, that's kind of what so I compared the frequency of size and age classes. The growth rate equation I'll go over in the next slide because it's another big proportion or big component of my study. For longevity, I had to calculate the average oldest age of a population. So longevity is kind of self-explanatory. Timing of maturity was compared by determining the size and age at which 50% of the population is reproductively mature. 2 condition indices were examined, Fulton's K, which measures how fat or healthy a fish is for its length. So looking at these two fish examples over here, if that guy's fatter, he'll have a higher Fulton's K and is healthier. And then I also use the hepatosomatic index for measuring condition, or HSI. And that's measuring, it's the ratio of the liver weight to the body weight. And that's because the liver is an indicator of, liver size is an indicator of energy reserve status. So, and then afterwards, I measured total mortality using an empirical equation that's based on maximum age, and total mortality, which is slightly different since it includes fishing mortality, and that's calculated using catch curve analysis. To compare growth parameters between populations, I use the two-parameter von Bertalanffy growth function. And so all of my figures will look pretty much like that, minus the cool fish. So this figure has age on the x-axis and length on the y-axis. And like many organisms, fish exhibit asymptotic growth, where it essentially stops growing larger at a certain age. And that's the equation above, but the only two parameters you really want to be looking at are L infinity and K. So L infinity indicates the maximum size of a fish, and K is the curvature coefficient, which indicates how quickly maximum size is reached. I also wanted to point out that L infinity and K are inversely related, so which means that if a fish is bigger, it'll take longer to reach that maximum size. But if it's smaller, it'll reach maximum size faster. So, which is why they're inversely related. And going to my results, I'll be presenting the results from the geographic-based differences first, and then going into the habitat-based differences after. So to orient you to this graph really quick, so from top to bottom, all the regions are organized in order of decreasing latitude. And on the x-axis, we have total length and age, respectively. And on the y-axis, we have frequency or count. The red bars in the middle here indicate median size. So by following those red bars, we can see that there's a trend, a latitudinal trend, where median sizes and ages decrease as latitude decreases, with the exception of Southern California, where these sizes and ages jump up a little bit. And comparing growth between regions, following that awesome rainbow pattern, you can see that L infinity is decreasing with decreasing latitude. And looking at the 95% confidence intervals for both parameters, the K coefficient down here, we can see that K is increasing as latitude is decreasing. Comparing between longevity, longevity decreases with decreasing latitude, except for Southern California, where, again, we saw those slightly larger sizes, so it jumps up again. And I use logistic regression to compare time at maturity, where size is on the x-axis and proportion matures on the y-axis. And this dashed line in the middle indicates proportion at 50% maturity. And again, we can see that size at 50% maturity decreases with decreasing latitude. But with age, there's no strong latitudinal trend in age at 50% maturity. And here we analyze condition. This one's a little bit more complicated. In order to control for the possible season and year effects, only lingcod caught in 2015 to 2017 outside of its spawning months were used for the subsequent analyses. And because I wanted to make sure that differences were driven by region and not size, I used residual analysis in the following condition ANOVAs. So we can see that there is no latitudinal trend in Fulton's K, which is the upper graph, but there is a slight latitudinal trend in the hepatosomatic index. But across both condition indices, we see that lingcod from Southern California has the highest condition. Looking at natural mortality, we see that there is a slight latitudinal trend where the northernmost and the southernmost regions are kind of opposite of each other, but the kind of mid-range regions are more variable. And since natural mortality is calculated using maximum age, it tends to be a little bit more sensitive to outliers. Total mortality was calculated using the Chapman-Robson method of catch curve analysis, and there's no latitudinal trend in this, but we can see that lingcod from Washington and Southern California have the highest total mortality. Going into some of the habitat-based differences we saw. So this is the same figure from earlier, except that the colors are switched around to represent habitat. So from now on, blue will be high relief areas, and the kind of gray will be a low relief area. And again, these bars here indicate median size, and we can see that lingcod from high relief habitats are consistently larger and older than lingcod from low relief habitats. And on average, the high relief individuals were larger by 21 centimeters and older by 2.9 years. Comparing growth, L infinity is higher in lingcod from high relief areas, but the K parameter is smaller. Lingcod from high relief habitats live longer, but they appear to reach 50% maturity at smaller sizes and younger ages. And here is the age plot. Lingcod from high relief habitats are in better condition across both indices, so they have more energy and they're fatter or healthier. And lastly, we saw that lingcod from high relief habitats have lower natural mortality, which makes sense since they get older, but they have higher total mortality, which is probably indicative of heavy fishing pressure. And my discussion. So I'll first go over a quick recap of what we found compared to what we predicted. What we found was, as latitude decreased, growth, longevity, mean sizes, and timing of maturity decreased, and natural mortality increased, which was consistent with what we predicted. However, we did see that condition increased, and there was no trend in total mortality. So let's look into some of the drivers behind the differences that we saw here. So it could be driven by environmental effects, such as patterns of sea surface temperature and productivity, which I will demonstrate more in Chapter 2. And looking at condition, I thought it was interesting that there was a latitudinal gradient in condition, but it was contrary to what we expected, given the California current. So we saw that condition was better in like southern warmer waters than it was in northern waters. This could be due to the fact that our collections were done during one of the strongest El Nino events in recorded history. So between 2014 and 2016, warm El Nino waters brought a northward shift in many marine species ranges, as well as a variety of unusual biological events. We found a lot of red pelagic crabs in the stomach from lingcod from Southern California, since they are opportunistic predators. So kind of that seasonal variability might account for why we're seeing this higher condition. In addition to environmental effects, there could also be fishing effects, which is typically called fishing-induced evolution. And this is because fishermen are typically targeting those big fish in a population. They're essentially removing large reproductive males and females. So studies have found that over time, individuals are smaller and they start to mature earlier at smaller sizes, most likely to maximize reproductive potential. However, even fishing effects on a population can be variable depending on a region's history of exploitation. So the most recent lingcod stock assessment reported that the exploitation of lingcod is considerably higher in California. The fishery started earlier also in California than it did in Washington and Oregon. So knowing these things about the fishery, it makes it difficult for us to really disentangle the factors behind what's driving these differences that we see. Is it coming from environmental effects or from fishing effects? So are growth rates faster and our sizes smaller in Southern California because of those warmer waters or because it was heavily exploited? So some of these fishing effects can be seen in our results from total mortality. And as a reminder, total mortality accounts for fishing mortality. But we would still expect to see a gradient of some kind in the total mortality figure from earlier, since Northern California and Central California were also fished at that time. So I believe that the significantly higher total mortality in Southern California is a product of the compounding effects of both fishing and environmental factors. And it kind of shows us that increased fishing pressure on a vulnerable population that has higher natural mortality rates might increase the total mortality overall. As for lingcod from Washington, because they have a lower natural mortality, they live longer, the high total mortality rate is most likely just from fishing effects. So looking at the habitat-based differences, again, a quick recap of what we found. So we found that lingcod from high relief habitats had faster growth, lived longer, were larger in size. They were in better condition, had lower natural mortality, and higher total mortality. And the only difference we saw was that we saw that they matured at earlier times than lingcod from low relief areas. So what are the drivers of the differences that we see here? So there could be habitat effects, such as differences in habitat quality, prey availability, provide a high quality environment that improves lingcod health and energy reserves, which could potentially lead to higher reproductive output and survivability. And looking again into the fishing effects, I'll do a quick recap. The recreational fishery for lingcod uses hook and line gear, targets high relief habitats, and the commercial fishery uses bottom trawl gear and targets low relief habitats. So it's important to note that fishing practices can change with time. In the past, the commercial trawl fishery was much larger than the recreational fishery, but within the past 15 to 20 years, it's shifted to the other way around. So essentially, the fisheries shifted from targeting low relief habitats to high relief habitats for lingcod. And this is reflected, this is reflected in the higher total mortality that we see in lingcod from high relief areas as well. And it might be the driver behind why timing of maturity is lower in lingcod from high relief areas, because they're fished heavier. So there's also the question about movement between the habitats. Since we know that juvenile lingcod will move from low relief into high relief areas when they get larger, and females will migrate inshore during spawning season. Since the majority of our sampling was outside of spawning periods, we weren't too concerned about migrating females. But as for the juveniles moving, we can actually see some of that movement in the size frequency histogram from earlier. So right around 30 to 50 centimeters, we can see that lingcod from low relief, the small ones, are kind of disappearing from that low relief habitat, and then they're reappearing in the high relief areas. However, you can still see some of those older, larger age classes in the low relief habitats. And because adult lingcod are relatively sedentary and have small home ranges, it's likely that some of those larger individuals that reside in those low relief habitats perhaps never move on to those inshore rocky environments. And again, the purpose of comparing the life histories between two habitats was primarily to highlight the fact that the lingcod stock assessment uses life history parameters based off of trawl caught samples only. So the populations that they're catching and what they're using for the stock assessments are those smaller individuals that are found in low relief areas. So now knowing that life history can be quite different in lingcod from high relief habitats, this tells us that stock assessment survey techniques should be reflective of both habitat types and both fisheries in the future. Some key points from Chapter 1 are that geographic and habitat differences in life history can be very prominent in broad-ranging species, and between environmental drivers and fishing effects, it might be kind of hard to disentangle the mechanisms behind what's really causing these changes. But fishery stock assessments need to address these differences in order to ensure proper management. And stock assessments using life history traits from only one region or one habitat can be an inaccurate representation of a population. So, but wait, there's more. So now that we've established that variability in lingcod life history exists, we can look a little bit deeper into the environmental correlates behind it and its implications for lingcod stock structure across the coast. So in this chapter, I'll be applying what we know about life history variability for use in informing management decisions. So a few studies have quantified the benefits to the fishery by using spatially distinct management options. One such study was conducted by Hamilton et al. Around the California Channel Islands. And in this area, the northern Channel Islands are exposed to the colder, nutrient-rich waters that come from the California current, and the southern Channel Islands are exposed to the warmer waters coming from the California countercurrent. So similar to in Chapter 1, this study found that geographic variation in sheephead life history was strongly linked to gradients in temperature, productivity, and fishing pressure in the Channel Islands region. And by using a population dynamics model, Hamilton et al. Found that by dividing the sheephead stock into two separate management regions that were based on life history differences, they were actually able to increase fisheries yield while still meeting sustainability goals. And I'll go a little bit deeper into the background of the lingcod fishery. So again, they are federally assessed on a regular basis. And I point this out because some stocks aren't assessed at all. So it's good that they are federally assessed, but there still might be things that are missing in the methods that they're using. So the lingcod are assessed as having a northern and southern stock, but this division is based on state line and not on biological parameters that are in the population. It was overfished in 2000, but was considered rebuilt coast-wide by 2005. However, the northern and southern stocks were rebuilt to different levels. So you can see that around 2005, while the northern stock is rebuilt to a little bit around the management target, if not past it, the southern stock is actually still kind of right below the minimum stock size threshold. And this is because there was and still is greater uncertainty regarding stock status in southern waters than there is in the north, and it's much more data limited. So this, but overall, this tells us that having broad coast-wide management might inflate stock productivity while missing out on local depletion. So the questions I was curious in asking for Chapter 2 are, what are the oceanographic drivers behind lingcod life history variability, and how can life history parameters be used to identify biologically significant breaks in lingcod stock structure? So it's currently managed as two stocks with the northern and southern stock as defined by state line, but is there a better location for that break, and is that the best number of divisions to be using? So I have two objectives in Chapter 2. Number one, I'm interested in investigating associations between life history parameters and how they're influenced by oceanographic indicators. And I predict that life history of lingcod will be directly related to latitudinal patterns we see in sea surface temperature and productivity. And here, chlorophyll A will be used as a proxy for primary productivity. My second objective is to identify biologically significant breaks in the coast-wide lingcod stock structure using life history parameters. And I predict that breaks in the population will be latitudinally driven and occur at major biogeographic boundaries, so at Cape Blanco, Cape Mendocino, and Point Conception. So for the purpose of looking at the more fine-scale patterns behind life history differences, I'll be addressing these two objectives on the port level. So while in Chapter 1 I use more broad regions and habitats, Chapter 2 will focus on using the ports themselves to discover trends in the population. All right. So principal components analysis was used to determine the association of lingcod life history traits and to examine the influence of sea surface temperature and chlorophyll A. Mean sea surface temperature and chlorophyll, oh my gosh. And chlorophyll A data was obtained from the NASA Earth Data Program for each port. To investigate geographic lingcod stock structure, I use a cluster analysis using Euclidean distances between ports. And a statistical clustering validation was used to determine the optimal number of clusters along the coast. A non-metric multidimensional scaling plot was used to visualize similarities in lingcod clusters, and a permanova or permutational analysis of variance was used to find statistical significance between these clusters. Results. So I'll first orient you to the PCA figure, the principal components plot, really quick since it's a little complicated. So PCA is a technique that's used to order multivariate data. So it takes a set of variables, and in this case, life history traits, and it reduces them into a smaller set of principal components that is still able to illustrate the important patterns in the data. So here are the life history traits that we used. And these vectors, the magnitude and direction of the vectors indicate how important each variable is to the component, and the direction of the vectors tells us how the life history traits are associated with each other. So for example, looking at longevity and natural mortality, since they're opposite from each other, it shows that they're inversely related. So in this PC figure, we have principal component 1, which explains almost 52% of the variability in the data. And PC1 has large positive associations with longevity, mean size, L-infinity, and age at maturity. And it has strong negative associations with K and natural mortality. So we can group these two, by grouping these variables together, we can say that PC1 is an indicator of size, age, and growth. And on the y-axis, we have principal component 2, which explains 25% of the variability in the data and has positive associations with condition and negative associations with age at maturity. So as condition increases, we find that size and age, or age at maturity, decreases. So PC2 is a measure of overall health. And by saving the principal component values, I then plotted each of them against mean sea surface temperature and chlorophyll A. So, and each point here represents a port. So we can see that there's a strong trend between PC1 and mean sea surface temperature, where size, age, and growth decreases as temperature increases. And size, age, and growth increases with increasing productivity. So this is consistent with the things we saw in chapter 1, where, or the latitudinal patterns that we saw, where older, larger fish are in those northern and colder waters that are more productive. And looking at PC2, there's no strong relationship between overall Lingcod health and sea surface temperature, but Lingcod health does increase a little bit with increasing productivity, though this relationship was not significant. And going into the cluster analysis, I was only interested in using life history traits that are commonly used in stock assessments, so I only looked at the growth parameters, maturity, and natural mortality. And we found that 2 clusters were optimal. This is a dendrogram for the clustering. And there's a lot of colors and labels going on, but basically want to see that the Lingcod stock does seem to break itself up into a northern and southern zone. So the purple and green ports here correspond to Washington and most of Oregon, and then these warmer colors, with the exception of Coos Bay, are associated with California. And the break here seems to be right around Cape Blanco. So we can better visualize the clusters in multidimensional space. And because MDS is a mapping technique, the axes here are somewhat arbitrary. So MDS analyses are unique because it preserves the ordering of relationships between objects or ports. So similar ports are plotted closer together, and different ports are farther apart. And in this plot, we're better able to see how the clusters are identified and how far apart they are from each other. And the life history traits in the middle are shown as vectors. And again, the magnitude and direction of the vectors show us how influential that life history trait is on determining those clusters. So we can see that the northern and southern clusters are primarily driven by differences in body size and growth. Oh, we missed this one. Okay, discussion and conclusion. So some key points for chapter 2. We see that life history is closely linked to oceanographic factors. Temperature and primary productivity appear to play a key role in influencing fish mortality, growth, and condition. We saw that a 2-cluster division in the coastwide stock as being optimal, with the split being right at Cape Blanco. So this is really close to where the current break is. However, like I mentioned before, the current stock assessment uses the boundaries based on state line because it was easier to regulate on the state level and easier to keep track of total landings. In the past, however, the Lingcod population was assessed on the coastwide scale, and it eventually was split up into a northern and southern zone in 1997, and this was due to drastic declines in the population. But even then, the separate stock assessments were conducted because the southern region was so data limited compared to the north, and not because of biological parameters. So even though we found that the division between stocks appears to be roughly at the same area, we now know that it's driven by differences in the population. And for future considerations, more research is needed. The analysis I conducted here still did not take those habitat effects into account, the reason being that more data would be needed on the port level. In chapter 1, all of my analyses were conducted on those broad latitudinal regions, and they still could be missing out on those fine scale differences. Finally, wrapping up both chapters, what do my results tell us about the future needs of Lingcod and other groundfish stocks? So in addition to the federal trawl survey, I would recommend the establishment of a coast-wide hook and line survey for Lingcod and other bottom fish species, so that biologically relevant life history parameters can be used in future stock assessments. So currently there is a hook and line survey that samples south of Point Conception, but not throughout the coast. And because the recreational hook and line fishery is so popular and widespread across the coast, our stock assessment methods need to reflect the life history of the populations that live in those areas. I'd also recommend a further investigation of the direct and indirect habitat effects on bottom fish communities. I thought it was interesting that Lingcod condition was so much higher in those high relief areas, with the caveat that condition is seasonally driven and can change annually as well. But still the question remains, how do habitat-based differences in bottom fish communities translate into health benefits, and how would this vary? And how would this affect the fecundity and reproduction for those economically important species? So the next step in this project would be to conduct a cost-benefit analysis of using localized management practices. Because, well, the purpose of this study is to promote sustainability and long-term productivity of fish populations, of course, but what are the more realistic socioeconomic effects on fishing communities? So from a cost-benefit perspective, will the fishing community be, how will the fishing community be benefiting from the localized management? And how difficult and costly would this be to monitor and enforce? So these are questions that would need to be addressed and quantified if regional management methods are to be adopted in the future. So all in all, there's much more to do, more lingcod to catch, other fish. Thank you, everyone. So I'll go into my acknowledgments. So I have a lot of people to thank, especially my thesis committee, and I'll go into them in the next slide. But particularly people from the Northwest Fisheries Science Center up in Seattle. When I spent my 4 months in Seattle, I worked really closely with Jamil and with Melissa Haltuck, who authored the most recent Lingcod stock assessment, and they really helped walk me through a lot of the stock assessment process. And of course, the rest of the crew that went to Puget Sound in Alaska, Greg Williams, Krista Nichols, Gary Longo, and Kelly Andrews. These guys were the fishing crew for those northern regions. And WDFW, I want to especially thank Robert Legoff, Jennifer Topping, and Sandra Rosenfeld. They were the ones that taught me how to process and age those fin rays. It's a long process. And these are the random places that we parked our RV in when we were sampling up and down the coast. Well, except for CCFRP. So those guys, they helped kind of nail down my sampling methods when we were out on the boat. So that was really, really useful going out with CCFRP in the early days. The FRAM division, where we got all of our trawl samples, all of these guys here they helped me organize meeting points where I can pick up like 200 pounds of frozen fish and put in my car. The CAP lab helped with aging. The shop guys here are amazing. I think I see Chris somewhere in the crowd. I gave them a very strange request, and they met that request. I said that I wanted a portable dissection table that could have railings on the sides to keep in fish guts, but also I wanted like a hole on each end so I can push the guts into a bucket. And they delivered pretty well. And they're like, all right, so you want that like Tuesday, or when do you want that? So those guys were really, really helpful, and I can go to them for any of those like more shop-related issues that I had. And of course, all of the MLML staff and admin and IT and the library folks that didn't mind if I was living there for weeks. And, and we had all of the Moss Landing volunteers that helped out on the fishing trips and for all of our dissection parties. Again, that. And I'd like to thank my thesis committee, especially thanking Rick for teaching me how to think like a fishery scientist and for always challenging me and making me think critically about my methods. Thank you, Gita, for supporting me and being the voice of reason sometimes. I was really glad to have been able to take the Baja class with you because I don't take too many classes, but working in the field is always like the best experience. And thank you to Scott and Jamil for making this project possible, for getting the funding, for making sure I wasn't super poor the entire time, and for always having your door open no matter how silly the question or if I had emotional issues. And also for teaching me the same stats over and over again sometimes, because I just would not grasp it. And it was great because when I was up in Seattle, I got to spend a lot of time with Jamil. So he kind of became my primary advisor for like 4 to 5 months. So I'd talk to him every day, and it was a fun change from Scott. It's just different. It's different. No, it's just,
He is much nicer than I am.
But I basically asked Jamil all the questions I was a little bit too scared to ask Scott. So it was good. The balance was perfect. And if you're wondering why Scott is my primary advisor, it's probably because he kind of beat Jamil to the punch there. I just want to point out, they sleep too, okay? It's not just me. All right, and so this is the Alaska and Puget Sound crew up in Seattle. We had a lot of fun. We have, oh my gosh, okay. Krista Nichols and Gary here, they're looking at genetic connectivity of the Lingcod stock across the coast. And they're tuning in right now, and they've been really, really helpful and just like really great to work with. We have our volunteer anglers, Brennan and Ken, who traveled up to Alaska to go fishing with us. And Greg Williams, who was great in organizing all the field logistics. And he's here also, eating healthy. And of course, we have Bonnie and my UREC interns, Kevin, Bailey, and Trevin. And I believe they're actually all here today, which is pretty awesome. I can't believe you guys showed up. Bonnie has been my rock. I remember when Angela Zepp defended, she said that everyone needs a Bonnie Brown on her thesis. And I was like, I have one. So yeah. But these guys, so Bonnie, Kevin, and Bailey were in the RV crew with me. And so we were basically stuck in a small RV for a month and a half together, and I'm very surprised they didn't get super sick of me. But we worked really long hours, and we'd spend a lot of time dissecting, and sometimes we wouldn't even have time to eat. But we found ways to make sure we could eat while dissecting. So, and of course, I'd like to acknowledge all of the boat captains and deckhands that we worked with, and all the volunteer anglers. This is our core group. I call them the core group because they would show up almost anywhere I asked them to. And Ken, who went to all of the states that we went fishing in. And it was just really amazing that I can call on you guys at any time, and you'd be there and catch fish for me. And hopefully now I can catch fish with you guys for fun and not research. And lastly, I'd like to thank my family. This is my dad up here who went fishing with me, and my cousin, and the rest of my Moss Landing family who came out on the boats, and as well as my cohort here, my Moss Landing family that kept me somewhat sane for 4.5 years. And also, special shout out to my dad again for being okay with me drying fins during Christmas on our kitchen counter, and then said that we have to get out of there, it smells like fish, and people are coming over soon. So yeah, thank you, Dad, for being patient. All right, and now I'm ready for questions. Matthew.
On the port analysis that you did in chapter 2, did you include the West Coast samples, or is that just Open Line samples?
Good question. I did include the trawl samples. So that was another reason why we excluded Puget Sound and Alaska from those analyses, because we didn't have any trawl samples from there, so we felt like it wasn't like a true representation of the population from both habitat types. Anne?
If you were able to eat any of your Lingcod samples, what was your favorite thing to cook with the Lingcod?
Just beer batter. Can't go wrong with beer batter. We did a taste test. I could tell you more about that later. Diana.
That was me. That was a great talk. I have a question about the deep sea, or the deeper populations, and what you do with those data and how it informs the fisheries models.
So you mean from like the trawl samples? Mm-hmm. So those I didn't use, so I didn't collect like every single Lingcod from the West Coast groundfish trawl survey. So I collected like a subsample, and they use that information anyways for their stock assessments. So I was able to collect like a subset of the data and use that information. So they use information from like all depth ranges from those trawlable areas. Any other questions?
Anyone else? All right.
burrito.